Okay, so let's wrap up chapter 12, the nervous system here, and talk about the final segment of our neuron. All right, we started off with the receptive segment, went to the initial segment, we just finished the conductive segment. We call this the transmissive segment because the neuron needs to transmit whatever signal or whatever message it's trying to set. If it's sensory input or motor output, all right, we have to devise a way to do that. And so we're going to rely on this structure right here, the synaptic knob. Okay? So let's continue the story. We generated our action potential. Right? It started off, we initiated it at the axon hillock, we propagated it, we kept it going all the way down the conductive segment of the axon. And it arrives here into the transmissive segment. So our action potential arrives, and now we have a new player in the game. Okay? We have voltage-gated calcium channels. So remember, the first word tells us what the stimulus or modality is going to be. So we need a change in the membrane potential. And then the other term in there, label, is calcium. That's what's going to travel through our channel once we open it. So the action potential is going to change the membrane potential. It arrives here at the voltage-gated calcium channel. The calcium channel opens up, and calcium is in a higher concentration outside the neuron than inside the neuron. Because at the synaptic knob, we also have calcium ion pumps. And their job is to pump calcium out of the synaptic knob. And so they created that concentration gradient. So calcium then diffuses into the knob. This is another reason why you'll understand why calcium is an important element to have in your body. You don't want to be low in calcium because then it affects our neuron function. Okay? So calcium moves into the knob, all right, and it's going to bind onto the synaptic vesicles. All right, so these synaptic vesicles are these kind of bubbles inside the synaptic knob that contain the neurotransmitter. And so they have these specialized proteins on the outside. The calcium is going to bind onto that. All right, and so it's going to turn the outside of those synaptic vesicles positive. Okay? And so those synaptic vesicles will then migrate or move towards the plasma membrane at the far end of the synaptic knob there, fuse with the synaptic knob membrane, and release, all right, the neurotransmitter via the process of exocytosis. That's when the cell expels things from the inside. That neurotransmitter will then cross over the synaptic cleft. It diffuses across the synaptic cleft. It binds onto the postsynaptic receptors there, okay? And then it will either activate or inhibit those postsynaptic receptors into doing something or not doing something, okay? Interesting thing, the neurons, this is one of my more favorite types of facts, can make more than one type of neurotransmitter, okay? But here's the problem. They can only release one type at a time, okay? And that's partially dependent on the frequency, the number of action potentials that come down to the synaptic knob, okay? That's kind of cool. They make more than one. They're not specific to just making one type of neurotransmitter. They can make several different types, all right? But they can only release one at a time. Okay? So again, you're going to see this repeat itself again when we get to muscles. Okay? Same type of phenomenon. We're going to review this concept. Okay? Action potential comes down. It stimulates the voltage-gated calcium channels into opening up. Calcium diffuses into the cell. All right? Calcium enters into the cell, binds onto the synaptic vesicles. The synaptic vesicles hold the neurotransmitter. The neurotransmitters, the synaptic vesicles migrate to the plasma membrane closest to the synaptic cleft. They fuse with the plasma membrane and release the neurotransmitter through the process of exocytosis. The neurotransmitter then diffuses across the synaptic cleft and will bind on to the postsynaptic receptors on the other side. So here's a nice picture. Okay, and this is The example here that we're showing you is what happens with muscles. Okay? So the neurotransmitter would be acetylcholine in this case. Okay, so you can see here our neuron comes down, it synapses onto these different muscle cells here. Okay, it hovers, it doesn't make physical contact, there's a little bit of space here. So the active potential travels down, it stimulates the opening of the voltage-gated calcium channels, calcium floods in, it will then bind on to the synaptic vesicles here, they'll migrate here towards the plasma membrane, fuse with the plasma membrane, and undergo the process of exocytosis where they release the neurotransmitter into the synaptic cleft. It diffuses across the synaptic cleft, and then it will then bind onto the postsynaptic receptors here. All right? And if there's channels there, then the channels will open up and we'll get some sort of action to occur. Okay? All right, so what is the sequence of events from the arrival of an action potential at the synaptic knob until the release of the neurotransmitter into the synaptic cleft? Pretty much what I just told you. Okay, the voltage-gated calcium channels open up, okay, because the action potential, calcium moves in, it binds to the proteins on the synaptic vesicles, and then those synaptic vesicles will fuse with the plasma membrane, undergoing that process of exocytosis, and release the neurotransmitter into the synaptic cleft. Very similar when we talk about chapter 10, and we're going to talk about the the neuromuscular junction there. Okay, so let's review. Let's look at some of the differences of graded potentials to action potentials. Okay, so graded potentials, all right, cause ion flow, all right, to cross the plasma membrane due to the opening of chemically gated channels. Action potentials cause the movement of ions across the plasma membrane because of the opening of voltage-gated channels. Okay, graded potentials can be either positive or negative changes, EPSPs versus IPSPs, whereas action potentials involve first depolarization, sodium coming in, followed by repolarization, potassium leaving. Right? Graded potentials are just that. They're graded, okay? So we can change, all right, the actual potential change, for example, going from that -70 resting membrane potential to a -72, all right? That's kind of a small change, all right, versus going from a -70 resting membrane potential to a -80, okay? But to do that, we just have to have a stronger stimulus. How do we get a stronger stimulus? More neurotransmitter. Opening of more chemically gated ion channels. Get more ions moving across the plasma membrane. Okay? Whereas an action potential is all or none. Okay? Once we reach threshold, boom, doesn't matter. It's not going to change the intensity. Graded potentials, we can change the intensity by making the stimulus stronger. Okay? Graded potentials are local. Okay? They're only going to occur in the dendrites and the receptive segment. Okay? And it's short, and it travels a short distance. Okay? Whereas action potentials will go all the way down the entire length of the synaptic knob. They will not decrease in intensity. Okay? Good to know those, the differences between the two. Okay? This is a very important fact, and I suggest that you definitely know this because I've seen it asked on tests before, because I've asked it. Okay? So, two things can affect the speed of a nerve signal, an action potential. All right? One is, and we just talked about this, if the cell or the neuron is myelinated or not. Okay? Saltatory conduction is faster than continuous conduction. And can you get more water to flow through a big, thick pipe compared to a small, tiny pipe? Yeah. Same thing with action potentials. Okay? So the bigger the nerve, or the diameter of the nerve, okay, the faster it conducts. All right? And that has to do with the amount of resistance. All right? I didn't cover this in class, okay? But it talks about resistance versus flow. And this is actually a concept that you'll get into more detail in 211, okay? So if we have less resistance, all right, we can have more flow, all right, or the speed of the flow of the action potential down the axon, okay? So keep in mind, if you are myelinated, all right, our signal will jump, not literally jump, but it moves faster, all right, between the nodes of Ranvier compared to that continuous sequential opening of those voltage-gated channels. Okay? So myelination and diameter will affect the conduction speeds of an axon. Okay? All right, a couple different types of fiber groups. It's important to know. All right, when we're talking about Group A, Group B, Group C. All right, Group A, these guys are the big dogs. You can see from their conduction speed, 150 meters a second. Man, that's fast. All right, so then we have to assume that these are the biggest, these are the fastest. All right, so they're going to have the largest diameter and they're going to be myelinated. All right, so this is nice to know. Most of our somatic sensory neurons, so most of the neurons that you're getting conscious input from, all right, are going to be Group A. All right, and all of the somatic motor neurons. So that means, all right, all the motor neurons that are innervating your skeletal muscle are Group A fibers. Okay, they're the biggest, thickest, fastest. All right, Group B are going to be smaller, 15 meters per second, which is still not too bad, trust me. Okay, but it's all right, 10 times less than a Group A. Okay, Group C, that's the smallest, 1 meter. Okay, so these guys will be smaller, obviously, in diameter, and they'll also be unmyelinated. All right, now if you look here, some of our visceral neurons, that's going to be the neurons that go to some of our organs. All right, things that are going to involve our unconscious perception. We won't have any idea. And some of our somatic sensory neurons from the skin. Now, here's an important thing. Your pain fibers, they go on Group C, which is good. There's a concept called the gate theory. I don't know if any of you have ever heard of it, but if you've ever banged your finger onto something, if you're hammering a hammer and you accidentally hit yourself and you start to shake your hand out, and it makes the pain less, it's because when you're shaking your hand out, you're bombarding your somatic sensory nervous system with all these sensations from the Group A fibers. And they're bigger and they're faster, so all of their sensory signals make it up to the brain, and it blocks the C fibers. They can't get in. They can't get on. So it blocks the C fibers from transmitting the pain signals. It's called the gate theory. Okay? If you've ever used a TENS unit, has anyone ever heard of a TENS unit? It's a pain thing. It's a little electrical stimulation thing. You put it on, and it uses electricity. It helps to decrease pain. It's based on the gate theory there. Okay? So fiber types, Group A, Group B, and Group C. Group A are the biggest. We'll revisit Group A when we do reflexes. Okay? And Group C are the smallest. Those are, and again, an example of Group C's are the pain fibers. Okay, pulse frequency, I talked about this briefly, all right, how many action potentials that you'll get per second, all right, and this is going to be something that is going to vary. Now, when we talk about, all right, impulse amplitude, that's going to be, you know, how big the wave is, all right, whereas frequency, it'll be how frequent the waves are. It's hard to draw this on with a mouse, I apologize, okay. Basically, what this has to do with, again, we're dealing with the stimulus strength here. And the stronger the stimulus, the more firing frequencies we'll see. Okay? We'll talk about this a little bit when we get into chapter 16 with the optic nerve here. But for example, bright lights. Okay? Bright lights, the photoreceptors in your eyes, all right? When we're dealing with bright lights, we are going to have a faster firing frequency than we would with dim lights. Part of that has to do with the difference in the photoreceptors, all right? But when we're dealing with the cones, the same type of photoreceptor, all right, we're just going to be stimulating because we're going to have a decrease in our stimulus strength with dim lights compared to bright lights, okay? We'll talk about this concept in chapter 10, all right, but when we're dealing with motor nerves, if we want to have an increase in muscle tension, the amount of force that we can generate for a contraction, all we need to do is increase the number of motor nerves that fire, all right, or we can increase, the frequency at which they fire. Just fire down more action potentials, okay? If we don't necessarily want to recruit more muscle fibers, all right? And then we can also deal with some of the neurotransmitters, all right? So the frequency rate, all right, the number of action potentials we fire, how frequently we fire them, can influence the type. Because remember, we talked about this, all right? A neuron can produce a couple different types of neurotransmitter, all right? And how we determine one type of neurotransmitter to be released over the other is the frequency of the action potentials. All right, that's one of the ways that we're able to vary the type of neurotransmitter that we're going to release. Has anyone ever had beef pot pies before? They're horrible for you, but sometimes they taste good. I have a craving for beef pot pies and scrambled eggs with cheese. I call that a garbage plate because literally you should dump it in the garbage before dumping it into your mouth. Anyways, it just jumped in my head. It's a bad thing to have cravings like that. Okay, so let's talk about neurotransmitters. Let's talk about the thing or the stuff that comes out of those synaptic vesicles. Yeah, it's a chemical, okay? But there's different types of neurotransmitters, okay? So let's talk a little bit about first, all right, our neurotransmitter, right? This is a chemical that's made by the neuron, and the neuron is nice enough that packages it up into these little envelopes, right, in the synaptic knobs. And they just wait there for an action potential to come along, all right, for calcium to stimulate the opening of the voltage-gated calcium channels, calcium to enter the knob. And then that will then stimulate the release of the neurotransmitter to diffuse across the synaptic cleft and then react with the receptor on the target cell. So it's kind of similar to hormones, all right? You know, the hormones are a chemical that are going to affect the target cell, okay? So neurotransmitters are similar like that, okay? And when they affect that receptor on the target cell, we're going to get some sort of response. That cell is going to do something. If it's a skeletal muscle, hopefully it'll contract the muscle, or a smooth muscle. If it's a gland, hopefully we'll get that gland to release whatever it's synthesizing, okay? So there's many, hundreds of different types, right, of neurotransmitters. We're going to cover just a few, okay? Some of the important ones here. Because it is important that you know the difference, because through the rest of this course and the second part of this course in 211, you're going to revisit some of these, not all of them, okay? So this is when we talk about the classification of neurotransmitters. So we have four main classes of neurotransmitters. The first one here, all right, is a very popular one, acetylcholine. Also, I write it down as, well, a lot of people do, ACH, acetylcholine, okay? And so a lot of times when we're dealing with neurons, action potentials, all right, the result or the physiological response will be what we call a binary one. It's either yes or no, 0 or 1, okay? Excitatory or inhibitory. Okay, so when we're dealing with the neurons, all right, we're going to play it as it's either going to be excitatory or inhibitory. So acetylcholine, all right, can be either or. And it depends on what it's interacting with, okay? If it's dealing with the somatic nervous system versus the autonomic nervous system. All right, when it's dealing with skeletal muscle in the somatic nervous system, it's always excitatory, okay? So when we get into the autonomic nervous system, it can be inhibitory. All right, the next class, all right, what we call the biogenic amines or the monoamines. All right, and so we have a classification of drugs that helps to block the synthesis of this type of neurotransmitter. Okay, called mile blockers. Okay, so you can see here, all right, we call it the biogenic amines. These are amino acids, right, that get slightly modified. All right, and we see the catecholamines, all right, dopamine. All of these, dopamine is a great neurotransmitter. Well, we'll talk. Nope, not going to talk about it now. All right, but the catecholamines, all right, are originally made from tyrosine, all right, which is an amino acid. All right, the indolamines, serotonin is an example. All right, that is made from either histidine or tryptophan. If you're familiar with tryptophan, is anyone familiar with tryptophan? That's right. Every Thanksgiving you become familiar with it. It's the amino acid that makes you drowsy. Okay? And so certain serotonin-esque drugs can do the same thing. All right, the fourth chemical class are just amino acids. We know that amino acids are protein building blocks. All right, but the common ones that we see are glutamate, glycine, and GABA. All right, they play an important role in our neurotransmitters. Mainly, several of these, the glutamate and the GABA, all right, we can see they play a role in our central nervous system. And then finally, the neuropeptides. All right, now these are not just amino acids, but they're chains of amino acids that we've strung along. And you see this whole list: endorphins, substance P, enkephalins, somatostatin. All right, somatostatin has to do with growth hormone. All right, endorphins, substance P, and enkephalins. Endorphins and enkephalins are that feel-good chemical that your body produces in a state of euphoria. So there was a movie that came out, I don't remember, it was in the 80s. It's called I Come in Peace with Dolph Lundgren. I don't know if you know who he is, but he was the Russian in Rocky IV. He's played in a couple different roles. But anyways, he plays this police officer that has to fight off this alien that came to Earth. And this alien was taking illegal street drugs and injecting it into people and getting them really high. And then he would siphon off their endorphins and their enkephalins. And he's basically an intergalactic drug dealer. And that's what he did. Dolph Lundgren had to hunt him down. Anyways, these guys are going to be made when you are in a state of euphoria. Substance P, unfortunately, is not made when you're in a state of euphoria. That's made when you're in pain. And substance P will irritate free nerve endings and send pain signals. Okay? Anyways, these are the four classes: acetylcholine, the biogenic amines, the amino acids, and the neuropeptides here. All right. So how we classify these neurotransmitters is by how they function. All right. So you have two types when we talk about how they affect the membrane potential. And I told you it's a binary thing when we're dealing with neurons. Yes, no, on, off, all right, excitatory or inhibitory. Okay. So if these guys are excitatory neurotransmitters, they're going to create EPSPs, which means they're going to open up the chemically gated cation ion channels. If they're going to be inhibitory neurotransmitters and generate IPSPs, then they're going to open up potassium channels and chloride channels. Okay? All right. Now, when we talk about how to classify them by their action, there's two types. They're direct and indirect. Okay? When we talk about direct, right, this is what we saw with the action of IPSPs and EPSPs. We release the neurotransmitter, it binds to a receptor, and that receptor then opens up that chemically gated channel. That's direct. Okay? Indirect involves secondary messengers. Okay? Now, I could go on for a while about secondary messengers, but I won't, because you will learn about that in much more detail in Chapter 17 when you're talking about hormones. But an example of a secondary messenger, I'll put it into perspective for you. First of all, it involves activation of what we call G proteins. Okay? So this neurotransmitter will bind to a receptor. It will cause the activation of the G protein on the inside of the cell. And then that G protein will activate a series of other enzymes to activate a specific substance to then maybe open up a gate or cause protein synthesis. All right? So the best way for me to describe this to you in an analogy would be like me coming to your door, you're upstairs folding clothes, you don't want to be bothered or doing whatever, taking a shower. and I knock on the door, and your butler comes down and is on the other side of the door, and I'm outside. He won't let me in, or she won't let me in. And I say, I have a message for you. I have to give Mr. or Mrs. So-and-so a message. And the butler's like, just tell me. I'll take the message to them. Okay? So that's pretty much what happens. I can't get into your house. The house is the cell. I can't get in. I'm the chemical neurotransmitter, and I leave the message with the butler. The butler then takes it up the stairs into the bedroom or into the study and gives you the message. And that's basically how it works for indirect transmitters. Okay? That involves our second messengery system. Okay? All right. So what are the four classes or primary classes of the neurotransmitters? Acetylcholine, amino acids, monoamines, and then the neuropeptides. So since we're going to be talking about acetylcholine in a little bit more detail in Chapter 10, let's just kind of give you a quick little brief understanding about it. Okay? All right. So when we talk about its use in the peripheral nervous system, it's going to be excitatory, or it'll stimulate skeletal muscle. All right. In the central nervous system, all right, it's going to change arousal levels here. So it basically comes from acetate and choline, okay? And it's synthesized from both of those compounds, and it's put together as acetylcholine, and it's stored in the synaptic vesicles. So we know the story from there. Those synaptic vesicles, when triggered, will release it into the cleft. It diffuses across the cleft, and it will attach to the postsynaptic receptor, okay, for a very short period of time, but long enough to create a signal, all right, to create some sort of action. So when there's no more action potentials coming down, and it's like having a party, you got to clean up. There's a mess everywhere. We got to clean up after the party. So the action potentials are done. All right, we have all this acetylcholine that's out in the synaptic cleft. Let's clean it up. Let's get rid of it. Okay. So we're going to clear it out from the cleft by breaking it back down into acetate and choline using this enzyme, acetylcholine esterase. Anytime you see a word and at the end it has ASE, it's an enzyme. You should know that. So anytime you see like kinase, acetylcholine esterase, can't think of any A's off the top of my head, protease. All right, anytime you see ASE in the end, it's an enzyme. So it's going to break something down, okay? So it breaks acetylcholine down into the acetate and choline, and then the cell, the presynaptic cell, takes it back up, all right? And it takes it back up, and it sticks it back together as acetylcholine back into the synaptic vesicles. Once it breaks it down into acetate and choline, it can no longer be effective. It's not an active neurotransmitter, and it won't be able to affect the target cell receptors anymore. Okay? So how it affects the target cell is going to depend on the type of receptors. And we have these two types of receptors. Again, I won't go into detail about the nicotinic or muscarinic, but those are, all right, some cell receptors. Okay? And when acetylcholine acts on either one of those types of receptors, right, you're going to have a varied or different response of the target cell depending on which of these receptors you stimulate or activate. Okay? Nicotinic are going to usually be excitatory, all right? Muscarinic aren't always going to be inhibitory, but they will have something similar to that effect depending on where we're talking about, if it's like in the heart or whatnot, okay? So just keep in mind, depending on the receptor will determine the effect on the cell, which effector it affects. All right, almost done, y'all. I'm going to wrap up with some more neurotransmitters here because, all right, there's a couple different ways. One of the ways that I just told you about to clear the synaptic cleft of a neurotransmitter was to have an enzyme that breaks that enzyme down. But there's a couple other ways that we can actually remove a neurotransmitter from the synaptic cleft. Okay, we saw the first one. We have an enzyme, breaks down the neurotransmitter, and it's gone, right? Another way to do it is what we call reuptake. Okay? That presynaptic neuron has this machinery, right, in its membrane to pretty much hoover up or suck up all of that neurotransmitter, right, back into the synaptic knob, right? And then in some cases, all right, that neurotransmitter just moves away from the synapse, and the neighboring glia take it in. So those are the three ways. Enzyme to break it down, okay? We have the reuptake transporters that will just hoover it up and suck it back into the synaptic knob, or it just moves out of the synaptic cleft and the glial cells take it up, okay? So there are drugs for certain conditions that will affect, all right, neurotransmitters. And in some cases, if we want to prolong the effect of the neurotransmitter, all right, we'll create a drug that's going to keep it in the cleft longer, okay? SSRIs, selective serotonin reuptake inhibitor. This classification of drugs prevents the serotonin neurotransmitter from being taken back up into the presynaptic knob, or synaptic knob, all right? Others will actually stimulate, all right, or inhibit certain enzymes from breaking down the neurotransmitter. All right, if you get an acetylcholinesterase inhibitor, all right, it will prevent acetylcholine from being broken down. It'll stay in the synaptic cleft longer. It will stimulate the post-receptor or the postsynaptic receptors longer. Myasthenia gravis is a condition in which they use acetylcholinesterase inhibitors. All right, myasthenia gravis is a muscle weakness condition. You have fewer receptors on the motor end plates, all right, and so even though you're releasing in a lot of neurotransmitter, all right, it doesn't connect very long. So by blocking the acetylcholinesterase, all right, enzyme from breaking down the acetylcholine there, we can keep it in the synaptic cleft longer. Also, we see it with Alzheimer's, right, when we're talking about the hydrobromide drug here. All right, so here's just a picture of those three phenomena occurring, okay? You can see here the acetylcholine being released, it goes across, all right, and then you can see how it has a direct effect. It stimulates, all right, this receptor here, and we'll have an EPSP, for example, occurring, or we have our secondary messenger pathway system here with the activation of the G protein, and then it causes a whole bunch of other chemical reactions further down the road here. All right, last part, neuromodulation. Okay, how we can regulate, all right, our signals here. Okay, so we have, all right, some chemicals that can affect the response of some of the neurons. And the two types of neuromodulation, all right, is facilitation and inhibition. So when we're undergoing facilitation, we're going to see an increased response in our postsynaptic neuron. Okay, so that could be, remember what I said, more is better. Increase the amount of neurotransmitter, okay, that's going to be found in the cleft, or in some cases, we can increase the number of postsynaptic receptors. Okay, increase the number of postsynaptic receptors means more ions can flow into the postsynaptic cell. Inhibition, all right, we're going to generate a weaker response. So we can then now decrease the amount of neurotransmitter, or we can decrease the number of postsynaptic receptors. Okay, facilitation versus inhibition. All right. All right, last part here. I don't know if you've ever heard of nitric oxide. All right, this is a wonderful, wonderful neurotransmitter. All right, helps for modulation of, and the nice thing about it too, I should really say this, is that it's short-lived. All right, And it was popular. I don't know if it's still popular with folks that like to exercise a lot, especially if they're trying to gain muscle mass. All right? But it helps because it, one, increases blood vessel dilation. If you increase blood vessel dilation, you increase blood flow. When you increase blood flow, especially if it's to a tissue that's undergoing high energy demands or high rates of metabolism, all right, this gas here will help, all right, to get those tissues that increase demand from the blood supply because we increase the flow here. All right, so we're going to see here, all right, with the nitric oxide, because it's short-lived and it's a nonpolar gas. Remember, if it's nonpolar, it has an easier time diffusing across the membrane there. And then this last one here, the endocannabinoids, we're seeing this a little bit more, especially as we get into the CBD oil, all right, derived from marijuana. Again, small nonpolar molecules. Well, we know we like that, especially if we're trying to diffuse across the plasma membrane here, all right. This one here has been great because we already know the effects of cannabis, all right. Definitely will help with appetite, and especially if you're dealing with terminally ill patients, those that are what we refer to as cachexic, and they're not eating as much, maybe because they're at the tail end of their cancer treatment. We give them some of this marijuana-esque drug, and it helps to increase the marijuana. It helps to increase their appetite. All right. Last slide. What is the term for the type of neuromodulation that results in a greater response from the postsynaptic neuron? Facilitation. In a lesser or weaker response, inhibition. All right. That is it for the nervous system chapter. All right. I wanted to wrap that up because I don't want to take away from muscles. I want to spend as much time on muscles as possible. All right, so let's just do some labeling here on the upper extremity muscles, and then we'll figure out what we're going to do here. Let me just